Hello, my name is Jeremy Chen, and I'll be presenting the novel discovery of a partiti-like virus explored through ecological and machine learning analyses. Just a bit of background first. One established order in biotaxonomy is Durnavirales, which hosts multiple families, including Partitiviridae. Within that, one genera is the cryspovirus. And what makes the cryspovirus so special is that it is the only one that infects protozoa, compared to its fellow genera that are known to only infect fungi and/or plants. Its only host is a protozoa, Cryptosporidium, and that is spread worldwide with all continents being affected, as shown in the red portions of the map. The protozoa has also been discovered in many species, including humans, birds, and bees, and we can infer that the cryspoviruses follow a similar geographic distribution and can be sampled from many species as well. The novel virus in question that I have termed as Durnavirus ambigiformis was initially found in a study that aimed to investigate novel stressors on honeybees in Australia, where a known stressor is absent. During the study, a novel pathogen was discovered in the RNA sequencing run. Since then, the virus has appeared in not only bees, but also in woodland mosquitoes and various aquatic environments such as wetland, river, and leaf metagenomes across North America and Australia. In its wide array of species and locations, it resembles cryspoviruses, but which could point to the idea that this virus could be a novel species. Following that, I hypothesized that this virus is a cryspovirus infecting a protozoan within the bee microbiome, and this can be measured by how similar it is ecologically and genomically to known cryspoviruses. Moving on to the methodology part, so I had two main forms of methods for the ecological side. So one was to investigate other samples that contain the virus, as well as viruses that are similar in sequence. So by investigating their metadata and similarity sequences, we can try to infer if any properties of our viruses contradict with those of the cryspoviruses. And the other one was to build a phylogenetic tree for Durnavirales that included my virus, and doing this would quickly give us an idea of whether it fits into one of the established families and genuses by observing the form clades. So for genomically, I want to assemble and analyze the RdRp genome, and through this we can verify whether or not our virus has the same length and number of open reading frames as a partitivirius should, and if it shares any conserved segments with known cryspoviruses. Secondly, machine learning analysis by using a pre-trained model that can pick up sequential and structural intricacies of the RdRp sequences, and we can then visualize to see if cryspoviruses are similar to the virus through clustering on a UMAP plot. So firstly, our ecological methods gave us evidence to reject the virus being a cryspovirus species. So when we compared the virus with known viruses, it matched to the black queen cell virus and the sackboot virus. And due to their very high coverage and residue identity stores, I infer that our virus may also share the same host range, only affecting honeybees. The singular sampling source contradicts with the diverse sampling of the cryspoviruses, which rejects the hypothesis. My phylogenetic tree also showed that the virus created a clade of its own, shown at the top right here, rather than joining any of the established partitivirius genera, including cryspoviruses. So this analysis also rejected the idea that the virus is a cryspovirus. Similarly, our genomic analysis also rejected in classifying the virus as such. So after assembling the genome, we found that the segment containing the RdRp, which is validated by discovering the conserved motifs A to C, to only be around 1.4 kilobase pairs long. So while this fits into the expected length of a general partitivirius, cryspoviruses have shown to be approximately 1.7 kilobase pairs in length in this segment, so this difference supports the rejection of the hypothesis. Similarly, our machine learning analysis had a similar result, where our virus did not cluster with cryspoviruses at all, but instead with Picobirnaviridae and partiviruses like gamma and delta, as we can see here on the bottom middle. So this overall just shows that a divergent virus species has been identified through these analyses that follow the same geographic distribution and large sampling stores like cryspoviruses, and this points strongly to the idea that this virus may be part of a novel genus within Partitiviridae, or that it may form a new family in Durnavirales. Either way, further exploring and classifying this virus is necessary to guide methods like antiviral drug designs in the case that this becomes or greatly strengthens pathogens that can affect the whole world in the future. And just a few takeaway points. Number one, Durnavirus ambigiformis does not fit into any of the established Partitiviridae genuses through ecological and genomic analyses. And then this points to the possibility that this is a novel family of the Durnavirales, or a novel genus of the Partitiviridae. And lastly, partiti-like viruses need to be explored further to understand their taxonomical classification to guide studies like preventive medicine. Thank you.